Yeah. So our plan is we will introduce you first, and then you give the talk. After you talk, there will be a panelist discussion, and then the audience will also join our discussion. It usually take 30 to 1 hour. It depends how many questions that we will have. I don't know whether fits your schedule or not.
Okay, great. So, how long do you want me to speak for? 30, 40 minutes or something like that to speaking?
Yeah, yeah,
Yeah,
around
okay,
40
that's
minutes.
fine. Okay,
Okay.
that's fine.
So I think we can start now. And
Okay,
then, yeah,
yeah. Do you want me to pull the screen down or leave it like this?
you can leave it like this.
Okay.
So good afternoon, Sidaf, and also good evening to friends in China and good morning to friends in the United States. Welcome to the 235th online forum of Metabolic Age Talk. My name is Li Heng Wang, an associate professor at Peking University Health Science Center. Joining me as a co-host today is Dr. Julie Bai. Uh, she's an assistant professor from UT Health, Houston. It's our privilege to introduce today's distinguished speaker, Dr. Sidaf Faruqi, a world-renowned British physician scientist whose pioneering work has revolutionized our understanding of the genetics and the neurobiology of obesity. Dr. Faruqi earned her MB CHB, which is equivalent to an MD degree, with honors from the University of Birmingham, followed by a PhD from the University of Cambridge. She then completed clinical training in diabetes, endocrinology, and general medicine. Currently, she serves as professor of metabolism and medicine at the University of Cambridge. She is also a fellow of the Royal Society and a member of the Academy of Medical Sciences. Her landmark discoveries on the melanocortin-4 receptor pathway and leptin deficiency have redefined research into appetite regulation and early onset obesity. To date, she has published over 180 papers in top-tier journals, including New England Journal of Medicine, Lancet, Cell, Nature Science, Nature Genetics, and Nature Medicine. Her work has identified critical genetic mutations linked to metabolic disorders, translating scientific breakthroughs into actionable clinic insights. In recognition of her extraordinary contributions to metabolic research, Dr. Faruqi has received prestigious accolades such as the Daly Medals, the Cornell Medal, which she shared with Dr. Steve O'Reilly, her mentor, and the American Diabetes Association Outstanding Scientific Achievement Award. Beyond her groundbreaking research, Dr. Faruqi is also a dedicated mentor who has nurtured numerous scientists now leading independent research programs or occupying key roles in academia and industry, focusing on bridging the gap between genetic discoveries and clinical applications in obesity treatment. Today, we also invited 4 panelists to lead our discussion after Sidaf's presentation, including Dr. Yongxu, who is a full professor from Baylor College of Medicine. Xu's lab aims to identify novel neural circuits, neurotransmitters, and intracellular molecules in the brain, which are critical for control of metabolism and behavior. Yong is also a collaborator of CDF. The second panelist is Dr. Zhang. He's a principal investigator from UT Southwestern Medical Center. His research focuses on identification of gene mutations that cause metabolic disorders in mice, and also investigations of their key functions in maintaining energy homeostasis in order to seek translational opportunities. The third panelist is Dr. Chaoping Wang, a professor from the pharmacology department and at Sun Yat-sen University. His lab is committed to uncovering novel mechanisms of obesity and developing transformative therapies by combining GWAS, animal and drug development studies. And another panelist is Dr. Jie Zheng, a principal investigator from Ruijin Hospital, Shanghai Jiao Tong University School of Medicine. His research is focused on investigations on the genetics of obesity and other metabolic disease. Finally, we would like to thank to the Nova Nordisk for its sponsorship for this distinguished lecture. Without further ado, please join me in welcoming Dr. Sidaf Faruqi. The title of her talk today is Lessons from the Genetics of Obesity. Dr. Faruqi, the floor is yours.
Great. Thank you very much for that kind introduction. Really great privilege to be able to talk to you all today. And I'm very excited to tell you about our science, and hopefully we can have a terrific discussion. So, of course, we know the problem of obesity is really a global problem, and increasingly so, of course, in countries like China. But across the world, the prevalence of obesity, defined using a body mass index more than 30, is rising. In the UK, 25% of adults have obesity. And in the US, of course, it's more than 30, even 35, 40% of people have obesity. And the reason that we worry about that is that obesity is associated with multiple health problems, as well as being a disease in its own right. So we know that people who have obesity have an increased risk of type 2 diabetes, high blood pressure, cardiovascular disease. Obesity is a major cause of both fatty liver disease and increasingly chronic liver disease and cirrhosis, and indeed the need for transplantation. In many Western countries, it's now overtaking alcohol as a major cause of chronic liver disease. Obesity also is a major driver of many cancers, and is increasingly likely to overtake smoking as a major cause of cancers. And obesity also increases your risk of neurodegenerative disease and certain types of dementia. As well as that, individuals with obesity have multiple joint problems, breathing problems, and as you can see, a whole host of other conditions which often affect their quality of life, their health, and their ability to work. Now, what's driving this rise in the prevalence of obesity across the world? Well, clearly what's changed in the last 30 or so years is our environment. And what we see is a real change in the amount of food that we have access to, the calories in that food, and its palatability, which means that we are very easy to overeat. Certainly, we eat more calories in general than we expend in physical activity. And as most people are now undertaking more sedentary work and lifestyles, this imbalance persists, and it's very easy for people to gain weight in this modern environment. However, it doesn't explain why within the same environment there's a lot of variability in weight. Some people are much more likely to gain weight than others, whereas some can eat what they like and they stay slim. And we now know that the reason for that variability is genetic factors. So there have always been some individuals who develop severe obesity, but it's very clear that our genes can influence weight right across the spectrum. As you can see from this photograph on the bottom right, showing identical and non-identical twins, and you see that the identical twins have a very similar body weight, body fat mass, and body distribution compared to the non-identical twins. So this suggests that genetic factors play a major role in weight. And in fact, the strongest evidence does come from twins who were separated at birth and live in different environments, where they still share a very similar body weight as adults. If you look at children who are adopted, they tend to have body weights that are very similar to their biological parents and different from the adoptive parents with whom they share the childhood environment. And some very critical studies were done by colleagues in Canada who showed that if you take identical twins and you keep them in a research study for a year and you overfeed them, the amount of weight that people gain is very similar for two members of a twin pair, but very different across the sets of twins. And together, this evidence shows us that a strong explanation for variability is genetic factors. And if you take any two people, 40 to 70% of the difference in their weight is down to differences in their genes. Now, what are those genes and how do they work? Well, actually, For most people, we're talking about a polygenic risk, and that means there are thousands of common genetic variants, which, when added together, give you an increased risk of obesity with increased BMI. And those genetic variants can affect your food intake, your basal metabolic rate, and even how many calories you burn with a fixed amount of exercise. We know that those common variants each have a small effect, and we can add them together to make a polygenic risk score. And people with obesity have a higher polygenic risk score. People who are thin have a very low polygenic risk score. They have less of the variants that increase their risk of obesity. That's why they're thin. There are also some rare genetic variants that have a major effect on weight, and that's really what I'm going to focus on now. And these are the genetic variants which are really, in a major way, disrupting the function of often a single gene, and that's enough to cause obesity. Now, specifically, if we focus on monogenic disorders, that's where there's a clear-cut link between a particular mutation and the function of a gene and a phenotype. And we and others have described many of those, and I'm going to focus on those today. There are also a bunch of chromosomal disorders which can often take out one or more genes and again cause a severe and penetrant phenotype. So where are these genes working? Well, we've known for 100 years, more than 100 years, that the hypothalamus in the brain is a key region for regulating appetite and weight and for maintaining homeostasis. And what that means is that as both our internal and external environment changes, we have to adapt to maintain things the same, maintain homeostasis, and the hypothalamus navigates that function by affecting our behavior, our hormone secretion, and autonomic nervous system outputs. And what we know is that in animals, if you fast them, and that they will obviously lose weight very quickly. If you release the fast, they eat quickly again to regain the weight and reestablish that trajectory. And this has really led to the point, the really seminal work from Jeff Friedman, which showed that a major molecular explanation for the regulation of energy homeostasis is the hormone leptin. And he found that the hormone leptin was missing in these mice, the severely obese OB mice, a natural strain, an inbred strain that develops severe obesity. Now, they lack the hormone leptin because they have a mutation in the gene that makes leptin, and if you give them leptin back, you can correct their severe obesity. So we know from the work of melanocortin. And it's really the POMC part of the system that we're really going to focus on, because these POMC peptides bind to second order neurons expressing the melanocortin 4 receptor to tell you to stop eating after a meal. Alpha MSH is an agonist of the MC4 receptor, and AGRP, made from these other neurons, is an antagonist of the same receptor. So this is a key control point for the regulation of eating behavior. Now, we first showed many years ago now that this pathway was relevant in human physiology through the study of two children who had severe obesity but had undetectable levels of leptin. Normally, because leptin is made from fat, the more fat you have, the more leptin you make. But these children had no leptin at all because they had a homozygous mutation in the gene that encodes leptin. So this was the first single gene defect causing human obesity, and really proof that leptin is necessary in human physiology. Now, we knew that giving the mice back leptin could correct their obesity. So we started the first clinical trial to give these children injections of recombinant leptin. And you can see from the photograph on the bottom left, the dramatic change in the same child who's then been treated with recombinant leptin. And by treating these children, we learnt about leptin's physiological role. And what we learned is that in the absence of leptin, the main phenotype is that these children are incredibly hungry. They always want to eat. They behave as if they're starving. And when we treat them with leptin, actually just after 4 days, you see a dramatic reduction in that food intake, and they start to turn away food for the first time. And we quantify that using an ad libitum test meal, basically a very big buffet where we invite the children to eat whatever they want. And they eat almost 5 times as much as controls to start with, but then they eat a lot less when they've been treated with leptin. Now, what we saw is that children and young adults with leptin deficiency don't go through puberty, but if we treat them with leptin, they go through puberty at the appropriate developmental stage. The same happens in animals, and it really demonstrates that leptin is a key signal by which weight regulation and the timing of puberty are linked together. Now, downstream of leptin, as I mentioned, the key peptide is POMC or pro-melanocortin, and that is cleaved into several other smaller peptides, including alpha MSH, which then bind to these melanocortin 4 receptor in the hypothalamus. And genetic disorders whereby you completely lose POMC or you lose the enzyme that cleaves the POMC will also result in severe obesity and multiple other features as listed here. Now, what we've learned over the years is that there are many other different genetic conditions which interestingly converge on the same pathway. And by studying 8,000 children with severe obesity as part of the Goose cohort, we've identified many of these genes. Here's just an example of one of those genes, which we studied with Young Chu, who's obviously a co-host today. And here what we showed is SRC1, which is involved in interacting with phosphorylated STAT3, a downstream mediator of leptin action, is a key modulator of leptin signaling. And humans with mutations in SRC1 have obesity. And here, Young's lab made a mouse model of a human SRC1 mutation, and we show that that mouse gains weight and develops severe obesity. And part of the mechanism is by affecting the firing of these POMC neurons. So that's a kind of interesting example of the different ways in which genes can affect the function of this key set of neurons. Another example is that several human conditions are caused by impairing the development of these circuits. So here's a family of 14 genes, ligands, receptors, and co-receptors called the semaphorin 3A family. And what these molecules do is they affect axon guidance. So in early development, your hypothalamic neurons need to know where to go. And semaphorin 3s play a key role in directing these neural circuits as they develop. And if you have a human mutation, we can show in cells and then here in a mouse that you affect the development of these neural circuits. And briefly, we also found that there are non-coding RNAs which can also affect the function here of the MC4R neurons in the paraventricular nucleus in work with Marcus Stoffel. So there's a whole bunch of different genes that can affect the development and the function of this POMC melanocortin system. Now, the melanocortin 4 receptor is a really key control point downstream of leptin in the brain. And we find about 5% of children in our cohort have mutations in this gene, which cause a loss of function. It's the commonest genetic form of obesity. About 1% of people with obesity have mutations, and about 2 to 3% of children with obesity have mutations. Now, here's a schematic showing this particular gene. It's a G protein coupled receptor, and that's been a lot of interest for developing it as a therapeutic. We've studied lots of different mutations in cells, and we find there's a clear, many different mutations can cause a loss of function. But what's very nice to see is that there's a link between the function of the mutation when tested in the lab and the clinical phenotype of patients who have these mutations. So the more severe the mutation, the more the person will eat at a test meal. So this evidence was really important in showing that MC4 is a critical regulator of human appetite and is also seen in mice. Now, one of the things that we've been interested in is whether we can try to understand more about how this system affects eating behavior, because there's a lot of evidence in mice that it not only affects the drive to eat, hunger, satiety, fullness, but other aspects of eating behavior. And here we followed up some studies that were done in mice where they had clearly shown that if you disrupt the leptin-melanocortin system, either genetically or pharmacologically, you increase fat intake and you decrease sucrose intake. Now, we did a study in humans where we provided people, lean people, people with obesity, and people with MC4 mutations, with different levels of fat content in their meal. And we did this by masking the meal so that people couldn't tell if it was low, medium, or high fat. And what we found is that people broadly ate the same, but the people with the MC4 mutations ate substantially more from the high fat meal. Now, we didn't know what was doing this because it was quite quick. It's too quick for it to be a gut hormone or gut signaling because the meal is finished within 30 minutes. So it must be something orosensory that is detecting the fat content and sending a signal in the brain to eat more. Now, we weren't sure if it's fat or if it's the fact that the food is rewarding. So we also did a sucrose test. And here we had low, medium, and high sugar food. And most people prefer things that are more sweet. But actually, the people with the MC4 mutations really didn't like the very sweet food. In fact, they often didn't want to do the test. And this was actually in keeping with the mouse data, where basically, if you lose leptin or the MC4 system, you get increased food reward, increased preference for fat, but not sucrose. And the reason for that is that leptin is the likely signal to defend against starvation. And so if you're starving, you're going to eat more, food is going to be rewarding, and you're going to want to eat more fat because it will allow you to survive longer. And of course, you get twice as many calories from fat as you do from carbohydrate. And also you can store your calories from excess fat much more easily, whereas you only have a limited ability to store carbohydrate as glycogen in the liver. So, of course, for many years, we watched but autism or Asperger's syndrome, impaired social interactions. And when we make mice models with these mutations, they also have both obesity and impaired social interactions or impaired social anxiety, really suggesting an interesting preservation of these behaviors across species. And another interesting group of phenotypes that we observed involves this behavior shown here, and that's aggressive behavior. So here we have a group of genes involving the BDNF pathway, brain-derived neurotrophic factor. Now, this is very widely expressed in the brain, but in the hypothalamus, it's highly expressed in the ventromedial hypothalamus. And BDNF is a neurotrophin. It's involved in the development, maintenance, and plasticity of neuronal circuits. So the children who have mutations in either BDNF tract B or SH2B1, an adapter molecule involved in BDNF signaling, have speech and language delay, autistic traits, and aggressive behavior, What do I mean by that? Well, here's a summary of some of the phenotypes we observed. And a number of these children, particularly the males, had quite aggressive behavior when they were children. It's hard to know if that is caused by the gene because, of course, there are many things that cause disordered behavior. But we then looked into the mice with my colleague Liang Yurui in Michigan, and he did a lovely experiment where he deleted SH2B1 in the brain. And you can see here the data from this resident intruder paradigm. So in this test, you have basically a mouse in a perspex cage, and then you uh, add another mouse or intruder mouse into that cage. And what you see is before long, the mouse will start to attack. So if you look at the knockout mice in the black bars, they don't take very long to attack. The frequency of attacks is very high, and the duration of attack is very high. They keep on attacking the other mouse. And when you correct the gene expression by putting SH2B1 back in the brain, you correct this behavioral phenotype. So very dramatic evidence demonstrating that the SH2B1 gene causes aggressive behavior when it's deleted from the brain in mice. Now, this made us think about a new gene that we recently worked on with Yong again, where we found some interesting insights into a whole spectrum of behaviors. Now, this gene involves a very highly conserved set of channels, and these are channels that are polymodal, so they're turned on by lots of different stimuli, including the activation of GPCRs and tyrosine kinase receptors. And these channels, the TRP channels or transient receptor potential channels, are involved in sensory signaling. So they're found in flies, in mice, in lots of different animals, and they affect things like temperature sensation, vibration, taste sensation, soy sauce, you can see here, garlic, mustard, chili peppers are detected by these different TRP channels. Now, we were interested in one particular TRP channel, and that's TRPC5, which is expressed on POMC neurons. Again, those favorite neurons in the hypothalamus that regulate appetite and weight. And previously, Kevin Williams and his lab had shown that these POMC neurons express the TRPC5 channel, and they are involved in the depolarization of these neurons in response to both leptin and serotonin. Now, we became interested because we found two children, family trees are shown here, who had a deletion on the X chromosome that takes out a single gene, and that gene is TRPC5. And when we studied those children, we saw that their clinical phenotype was quite severe and quite distinctive. So they had intense food seeking and food hoarding behavior. They had severe anxiety and autism. They had maladaptive or aggressive behavior in response to sensory stimuli. They would often stay awake, often for 2 days even, and their mothers also had postnatal depression. We then found a bunch of missense mutations in the same gene in our goose cohort, and with Yong's lab, we studied these in cells, and we could see that many of these mutations cause a loss of function in cells. Now, still, it remains challenging to be able to link this particular receptor to those phenotypes. And there, again, we worked with Yong to make a mouse model of the human mutation. And what we found is quite remarkably, the male knock-in mice display many of those phenotypes. So here we can see that they have an increase in food intake. Interestingly, they also display food hoarding behavior, particularly when the temperature is altered. And Yong's lab showed that most of this effect is mediated by POMC neurons in the hypothalamus, as we would expect. What we also saw is that these male mice exhibited the same behaviors as we saw in the male patients. So they had marked anxiety. You can see this open field test. The knock-in mice, they hang around the outside. They're very anxious. They don't want to kind of explore. They're relatively aggressive, and they attack for longer. They tend to stay awake. And interestingly, in the social interaction test, they would rather interact with an object than they would with another mouse, which is very unusual behavior. So remarkable conservation of these behaviors in the mice that carry the human mutation in TRPC5. Now, uh, given this preservation, we wondered really whether some of the female behaviors could be preserved. But I did feel slightly strange when asking Yong whether he could look at this, because it was unusual to think about the behavioral phenotype in the women, in the people, because the mothers had postnatal depression. It's hard to say what that might look like in a mouse, but we said, could we just look at the behavior of the mothers? And what Young's lab found is that actually the mothers with the TRPC5 mutation display very abnormal maternal care behavior. So here's the wild type mouse, which normally would make a nice, neat nest and put the pups back into the nest. But here, the mouse with the mutation makes a very disordered nest. It's a real mess. And then sometimes this mouse will pick up the babies, sometimes it will leave them where they are, and it really does not care for them very well at all. Very disruptive maternal care behavior. And then we can quantify that behavior as shown here. It takes a long time for these mothers to pick up the pups. They often don't gather them completely, and they spend much less time with them. And we also looked to see if there was an effect on depressive behavior, and indeed, Young's lab showed that these mice have a greater immobile time in the forced swim test, and they also have reduced sucrose preference, key measures of depressive-like behavior in animals. And interestingly, this was only seen in the mothers, not seen in virgin females or in males, so only in this postnatal period. What could be driving it? Well, one of the features could be a reduction in prolactin. Normally, mice, the prolactin goes up on suckling, but here there's a marked reduction in the mice when it goes up on suckling. And the other thing is that these TRPC5 neurons are co-expressed with oxytocin. As you can see on this graph over here, high levels of co-expression in the paraventricular hypothalamus. And so then we deleted TRPC5 from oxytocin neurons. We can recapitulate all of those phenotypes in the male mice and indeed in the female mice, again with this impaired maternal behavior and also depressive behavior, including reduced immobile time, increased immobile time, and reduced sucrose preference. So the key experiment, of course, is to then put the TRPC5 expression back into those oxytocin neurons. And then when Young's lab did that experiment, they could reverse all of the phenotypes seen in these mice, proving that it was the TRPC5 acting on oxytocin neurons that was really mediating this maternal care behavior. Okay, so how do we explain all of these things together? Well, what's really fascinating is that there are a lot of studies already showing that these behaviors are tightly linked in animal models, in flies, in mice, etc. You have eating behavior, you have social interaction, you have reproductive behaviors, you have aggression and maternal care. And they're linked because they're all fundamental for survival. They're innate behaviors that are hardwired in the brain, and they involve neural circuits that lie between the hypothalamus and other brain regions as shown on this map. And essentially what's happened is we have found loss of function mutations in key molecules that affect the development, the function, or the maintenance of these circuits. And as a result, we see hyperphagia, obesity, and maladaptive behaviors. So together, this work really provides us with an insight into the biology of something very fundamental, and that's our innate behaviors in mammalian species. So to finish, I hope I've convinced you that obesity is complex, it's an interesting problem to study, it's clearly important at a societal level, and it arises from the interaction of social factors, the environment, and genetic factors. Now, those genetic factors operate in all of us, not just in people with obesity. The people with obesity or severe obesity, the genetic factors are playing a much bigger role in those individuals, but they act right across the spectrum, and they also explain why some people remain thin. Now, those genes likely affect many processes, but our work to date has shown that the principal driver of obesity is a failure of the control of appetite. And interestingly and importantly, human appetite is not just under voluntary control. People have often thought it's just simply up to the person to decide how much they eat. But our work has shown that eating behavior and appetite has a strong biological basis, as an innate behavior, and also, of course, there is a learned component that comes on top. And together, we try to use these genetic studies to provide insights into mechanisms and identify and validate targets for weight loss therapy. So this work has been formed clinical management. So clinical guidelines globally now recommend genetic testing as part of the evaluation of children with severe obesity. This work provides the rationale for treating both children and adults with severe obesity. There are a number of treatments. Leptin, I mentioned, MC4R agonists, and there are a number of treatments that are being developed in what is now a very exciting field. Importantly, by demonstrating the biological basis for obesity in patients, it helps us to remove the weight stigma from families. And that's particularly important to also remove the bias that comes from healthcare professionals, because this gets in the way of us supporting and treating people with obesity. And finally, there's just some images here, and this is just a reminder to me about this particular headline from newspapers in England about a child with obesity who sadly died at the age of 3, but the parents were blamed for causing that child's obesity when actually the child was a patient of mine who had a leptin receptor mutation. And this is a reminder really that as both clinicians and scientists in the field of obesity, we have an obligation to communicate that science so that people can understand that obesity is not simply a choice, but it's a biological condition caused by a disruption of the normal mechanisms that regulate weight. So I'm going to finish by thanking my team, our many collaborators around the world, our funders, and most importantly, the patients and their families. Thank you very much.
Thank you, Siddharth, for the great talk and the comprehensive insights about the studies of human genetic obesities, and also like how do you push it to the clinic management. So now let's invite our panelists. So the first panelist is Dr. Yongxu from Baylor School of Medicine. So I think Julie, please take over. So yeah.
Yeah,
Go
sure.
ahead.
You go ahead.
Hi, Sarah. Good to hear you again from this forum. I'll just throw out a few questions. Some of them we actually discuss in different occasions, but for the general audience here, I think it's important to discuss this as well. So the first